Microbial genetics. Microorganisms were generally ignored by the early geneticists because they are small in size and were thought to lack variable traits and the sexual reproduction necessary for a mixing of genes from different organisms. After it was discovered that microorganisms had many different physical and physiological characteristics that are amenable to study, they become objects of great interest to geneticists because of their small size and the fact that they reproduce much more rapidly than larger organisms. Bacteria became important model organisms in genetic analysis, and many discoveries of general interest in genetics arose from their study. Bacterial genetics is the center of cloning technology. Microbial genetics had its origin in the 1940s and 1950s as researchers such as George Biddle and Ed Tatum developed biochemical genetics through studying mutants of Neurospora. The genetic study of microbes has played a highly significant role in the recent developments in molecular biology, recombinant DNA technology, and the preparation of useful products such as insulin, human growth hormone, and blood clotting factors. It was no coincidence that the first artificially produced hybrid DNA was constructed using bacterial plasmids, and many of the spectacular advances and discoveries had been dependent on microbial systems or on microbial models. This success can be traced back to the first experiments in the molecular genetics of DNA in the genetic transformation of bacteria, as well as the first isolation of metabolic mutants in fungi. Microbes are ideally suited to the combined biochemical and genetic approach, which had early successes in the solution of the genetic code and the regulation of gene activity. The discovery and analysis of plasmid and the bacteriophage systems laid the foundation for the exploration of recombinant DNA techniques, which in their turn were dependent on the discovery of highly specific enzymes also in bacteria. These techniques have revealed details of genetic organization which traditional genetic methods could not have brought to light. Microbial genetics is a branch of genetics concerned with the transmission of hereditary characteristics in microorganisms. Within the usual definition, microorganisms include prokaryotes like bacteria, unicellular and mycelial eukaryotes, example yeast and other fungi, and viruses, notably bacterial viruses or bacteriophages. Microbial genetics has played a unique role in developing the fields of molecular and cell biology, and also has found application in medicine, agriculture, and the food of pharmaceutical industries. Because of their relative simplicity, microbes are ideally suited for combined biochemicals and genetic studies, and have been successful in providing information on the genetic code and the regulation of gene activity. The operant model, formulated by French biologist Francois Jacob and Jacques Monod in 1961, is one well-known example, based on studies on the induction enzymes of lactose catabolism in the bacterium Escherichia coli. The operon has provided the groundwork for studies on gene expression and regulation, even up to present day. The many applications of microbial genetics in medicine and the pharmaceutical industry emerge from the fact that microbes are both the cause of disease and the producers of antibiotics. Genetic studies have been used to understand variation in pathogenic microbes and also to increase the yield of antibiotics from other microbes. Hereditary processes in microorganisms are analogous to those in multicellular organisms in both prokaryotic and eukaryotic microbes. The genetic material is DNA. The only known exception to this rule are the RNA viruses. Mutations, heritable changes in the DNA, occur spontaneously, and the rate of mutation can be increased by mutagenic agents. In practice, the susceptibility of bacteria to mutagenic agents had been used to identify potentially hazardous chemicals in the environment. For example, the ARMS test was developed to evaluate the mutagenicity of a chemical in the following way: plates containing a medium lacking in, for example, the nutrient histidine, are inculcated with an histidine-requiring strain of the bacterium Salmonella typhimurium. Thus, only cells that revert back to the wild type can grow on the medium. If plates are exposed to the mutagenic agent, the increase in the number of mutants compared with unexposed plates can be observed, and a large number of revertrants would indicate a strong mutagenic agents. For such studies, microorganisms offer the advantage that they have short mean generation times, are easily cultured in a small space under controlled conditions, and have a relatively uncomplicated structure. Microorganisms, and particularly bacteria, were generally ignored by the early geneticists because of their small size and apparent lack of easily identifiable variable traits. Therefore, a method of identifying variation and mutation in microbes was a fundamental for progress in microbial genetics. As many of the mutations manifest themselves as metabolic abnormalities, methods were developed by which microbial mutants could be detected by selecting or testing for altered phenotypes. Positive selection is defined as the detection of mutant cells and the rejection of unmutated cells. An example of this is the selection of penicillin-resistant mutants achieved by growing organisms in media containing penicillin such that only resistant colonies grow. In contrast, negative selection detects cells that cannot perform a certain function and is used to select mutants that require one or more extra growth factors. Replica plating is used for negative selection and involves two identical prints of colony distributions being made on plates with and without the required nutrients. Those microbes that do not grow on the being made on plates with and without the required nutrients. Those microbes that do not grow on the plate lacking the nutrient can then be selected from the identical plate, which does contain the nutrient. Importance of microbial genetics. First, to understand the gene function of microorganism. Second, microbes provide a relatively simple system for studying genetic phenomenon and thus useful to other higher organisms. Third, microorganisms are used for isolation and multiplication of specific genes of higher organisms, which is referred as gene cloning. Fourth, microbes provide many value-added products like antibiotics, growth hormones, etc. Microbial genetics will be helpful to increase these products' productivity by microbial technology. Fifth, understanding the genetics of disease-causing microorganisms, especially virus, will be useful to control diseases. Sixth, gene transfer among the prokaryotes play a major role in the spread of the genes in a particular environment. Microbial genetics will be useful to study the gene transfer from one organism to another.